Hello students, the next point in your syllabus, that is the DNA replication in eukaryote. So in last point I already discussed about the DNA replications. Then there is the DNA replications in prokaryote. So today we move towards the DNA replication in eukaryotes. So in introductory part or the question asked in exam for write down the process of eukaryotic DNA replications or explain the role of various enzymes involved in eukaryotic DNA replications. For introductory part, you have to write down about the replications, the definition of replication, then how the replication takes place in the eukaryotic cells. So you have to only mention about the eukaryotic cells. So in the eukaryotic cells, mostly DNA replications takes place in the nucleus, and in case of prokaryotic cell, the replication takes place in the cytoplasm. So in the eukaryotic cells, there is the multiple sets or multiple sites of the origin of replications. So in that, these DNA replications carried out in 3 different steps, same like the prokaryotic cell. First stage or step is the initiation, second step, that is the elongation, and third step, there is the termination. So some quite complex process is observed in the eukaryotic cells. In prokaryotic, it is very simple and very few enzymes are used in the DNA replications as compared to the eukaryotic cells. In eukaryotic cells, there are somewhat quite complex process. That's why you have to understand step by steps. So in the initiation stage, first there is the binding of 6th subunit complex to the DNA strands. तो पहले क्या होता है, जो एक 6 सब यूनिट का एक कॉम्प्लेक्स फॉर्म होता है और वो कॉम्प्लेक्स DNA स्ट्रैंड्स को बाइंड होता है बिफोर द रेप्लिकेशन स्टार्टिंग. सो दिस इज द इनिशिएशन स्टेप. इनिशिएशन स्टेप में क्या होता है, जो 6 सब यूनिट कॉम्प्लेक्स तैयार होता है और वो कॉम्प्लेक्स DNA स्ट्रैंड्स को अटैच होता है. तो ये 6 सब यूनिट कॉम्प्लेक्स क्या है, तो वो हम देखेंगे कि 6 सब यूनिट कॉम्प्लेक्स में एग्जैक्टली क्या क्या है वहाँ उसमें. तो पहले क्या होता है, दिस ओरिजिन रेप्लिकेशन कॉम्प्लेक्स, दैट इज द ORIC. This ORIC binds to the origin of replication. Then there is the activation factors are formed. So one is the replication activator proteins and replication licensing factors. So these 2 factors are produced and they are attached to the ORIC regions. So the set of 6 microchromosomal proteins binds to origin of replications. पहले क्या होता है, जो 6th माइक्रोक्रोमोसोमल प्रोटीन्स है, माइक्रोक्रोमोसोमल प्रोटीन्स जो ORIC रीजन है, ओरिजिन ऑफ रेप्लिकेशन रीजन है, उससे अटैच होते हैं. देन फॉर मिनी क्रोमोसोमल, दैट इज microchromosomal 2 to microchromosomal 7 are binds and they forms the microchromosomal proteins. That means in case of prokaryotics में हमने देखा था जो DNA B जो प्रोटीन्स है, सेम लाइक यहाँ पे माइक्रोक्रोमोसोमल प्रोटीन्स जो DNA स्टैंड है और जो ORIC रीजन है ORIC regions को अटैच होते. These mini chromosomes causes unwinding of DNA at the replication forks. Then these single stranded DNA binding protein, which is called as a replication protein A, that are also binds to the strands. So these strands are always stay as a or they always keep them in a separated forms. तो इन द इनिशिएशन स्टेज आप फिगर में देख रहे हैं. तो डीएनए कहाँ पे प्रेजेंट रहता है, तो जो न्यूक्लियस है, न्यूक्लियस के अंदर जो क्रोमेटिन मटेरियल्स रहता है उसके अंदर डीएनए प्रेजेंट रहते हैं. सो दिस डीएनए in eukaryotic there is the number of replication sites. So in that there is the origin of replication, that is the ORIC region. So in that ORIC region first attach the there is the sixth subunit complex, that is the sixth microchromosomal proteins. Then these microchromosomal proteins activated this ORIC region and they forms the replication activation protein, that is RAP, and replicating or replications licensing factor, that is the RLF. Then these two regions, that is RLF and RAP, that means the licensing factor and activator proteins attached to the ORIC region, and these whole are form the pre-replication complex. As well as in the initial stage, there is the addition of the SSP proteins, or there is the also name as the replication promoter or the activator proteins, or a replication protein A. So these replication protein A or SSB can work as or they can add for the separating the two DNA strands. So you have to see in the figure. First there is the addition of MCM proteins, that is microchromosomal proteins, and these proteins are make separated the two DNA strands. Then there is the addition of SSB proteins or the replication proteins A. These replication proteins make the two strands separate. This is the initiation stage. Then in elongation stage, in this elongation case there are 5 different polymerases, that is alpha, beta, gamma, delta and epsilons are used. तो जो न्यू डीएनए स्टैंड्स इलोंगेशन में क्या होता है कि न्यू डीएनए स्टैंड्स फॉर्म होता है. तो यहाँ पे फॉर elongation or elongating the new DNA strands, 5 different types of polymerases are used. So first there is the primers. So DNA polymerase alpha in association with the primase forms the RNA primers. तो पहले इलोंगेशन के लिए क्या जरूरी है, वहाँ पे प्राइमर्स आरएनए प्राइमर्स ऐड करना है. आरएनए प्राइमर्स, these RNA primers contains about 20 न्यूक्लियोटाइड्स. उसके अंदर 20 न्यूक्लियोटाइड्स रहते हैं. सो दिस इज द जो प्राइमर्स है, प्राइमर्स आर द स्टार्टिंग और initiates the new strands in the direction of the 5' to 3'. So first enzyme that is used in the elongation, that is the polymerase alpha. Then DNA polymerase delta synthesize the lagging strands, and DNA polymerase epsilon synthesize the leading strands. So for synthesizing the DNA strands, continuous or discontinuous. So this lagging, that means the continuous strands is formed by the DNA polymerase delta, and for forming the leading strand, that is the discontinuous strands, the DNA polymerase epsilons are help to forming the leading strands. So this DNA polymerase delta and epsilon requires the sliding clamp. That means these for proliferating cell nuclear antigens. Then these proliferating cell nuclear antigens keep the DNA poly firmly on the DNA. जो डीएनए सिंथेसाइजिंग का काम करने के लिए वहाँ पे कुछ एंटीजेंस भी ऐड होते हैं, दैट इज द प्रोलिफरेटिंग सेल न्यूक्लियर एंटीजेंस. Then in last there is the supercoiling removed by the topoisomerase enzymes. तो जो due to the new strands formation, there is some trace are occurring on the remaining part of the DNA. So for removing the trace of this DNA, the topoisomerase are used. Then you have to see in the figure, there is the two strands, they are from forked, and at the one side there is a continuous strand, there is that is leading strand, and the discontinuous strands are observed, that is the lagging strand. So these the new strands are forming from the additions of the primers, or the new strands are formed from the primers at the 5' ends or the 3' ends. So the last step is the termination. In termination, at telomere regions of each of the newly synthesized strands, there is a RNA primers at 5 end. So after RNA primers are removed, DNA polymerase can't fulfill the gap because the enzymes can't initiate new DNA synthesis. If this gap not filled, chromosome become starters. So enzyme telomerase, that is composed of protein and RNA complementary to the telomeres, binds to these telomeres and catalyzed the addition of new repeats. Then these telomerase catalyze the synthesis of a new telomere repeats using the RNA templates. After these enzyme telomerase is removed, the new DNA is synthesized on the templates starting with an RNA primers. Then after the primer is removed. So in case of the termination, there is at the one side there is the lagging side. So lagging, that means the some gaps are present in the some gaps are present in new synthesized DNA. जो नया DNA synthesize किया है वहाँ पे कुछ gap रहते हैं. So in that case, the enzyme telomerase is work on that. This enzyme telomerase produces the templates. So these templates are synthesize the new DNA repeats. Templates क्या करते हैं, वहाँ पे new DNA repeats वहाँ पे form करते हैं, and then these telomerase is join this all the DNA strands, and then there is the removed the templates and the primers, and they add this. Then after the removing, there is the continuous new DNA strands is formed. So this is happening in the termination stage. टर्मिनेशन में यहाँ पे क्या होता है कि जो जो लैगिंग स्ट्रैंड्स है, मतलब जो डिसकंटिन्यूस स्ट्रैंड्स है, वहाँ पे जो यह एंजाइम टेलोमरेज वर्क करता है। वो क्या करता है, कुछ प्रोटीन्स और आरएनए कॉम्प्लीमेंट्री वहाँ पे प्रोड्यूस करता है। प्रोड्यूस करके वहाँ पे जो टेम्पलेट्स की मदद से वहाँ कुछ न्यू रिपीट्स वहाँ पे क्रिएट्स किए और सिंथेसाइज किए जाते हैं। देन दे कैन बी जॉइन द ऑल डीएनए स्ट्रैंड्स एंड दे आफ्टर द जॉइनिंग दीज telomerase or templates or primers are removed from these regions, and there is the continuously two new DNA strands are formed. So this is happening in the termination stage. So this is all about the replications in eukaryotic cells. So lastly I समराइज दिस रेप्लिकेशन। यहाँ पे आपको क्या क्या याद रखना है, तो इसमें जो रेप्लिकेशन डीएनए रेप्लिकेशन इन यूकैरियोटिक्स में मोस्टली टेक्स प्लेस इन 3 steps. फर्स्ट इनिशिएशन, elongation and termination. So in case of elongation, first there is the binding of 6th subunit complex. That means the 6 microchromosomal proteins are produced, and these microchromosomal proteins are bind to this ORIC region. So in case of eukaryotics, this replication takes place in the nucleus, and there are many sites for the origin of replications. So after the binding of 6 microchromosomal proteins, they forms the replication activator protein and replication licensing factors, and they combinedly called as the pre-replication complex. So these microchromosomal proteins that are equal to the DNA B protein, which is observed in the E. coli. Then in that initiation stage, there is some addition of the SSB protein, that is single stranded protein, or the replication protein A. You have to see in the figures, there is the replication forks are formed, and at that point there is the macrosomal proteins and SSB proteins or RPA proteins are binds to these two separated DNA strands. Then in the elongation stage, there is the function of 5 different polymerases, that is alpha, beta, gamma, delta and epsilon. In that case, the primers DNA polymerase alpha in association with primase forms the RNA primer. So this RNA primer starts to initiate new strands in 5' to 3' direction. Then in the synthesis of new strands, there is the functions of DNA polymerase delta and epsilon, so they can synthesize the lagging and leading strands. Then there is also addition of the enzyme topoisomerase, so they can remove the topological stress. And at the last step, there is the termination. In the termination, some in case of the lagging strands or discontinuous strands, some gaps are present. In that case, these enzyme telomerase are functions, and they can bind to the, or they can mix the new repeats, and they join to the all discontinuous DNA strands, and they form newly two two DNA strands. And after the forming of two continuous DNA strands, they removed the primers, that is the RNA primers on the both ends, and in this way the replication process will be takes place. So I hope this is somewhat complicated process, but at the view of exam, this is important question. So I hope all of you are understand this eukaryotic DNA replications.